Hello everyone, and welcome to the course of Comprehensive Molecular Diagnostics and Advanced Gene Expression Analysis. So, this is the week 5 discussion, and I am Udiptya from IIT Madras. Okay. So, this week's videos were primarily on DNA sequencing. It started from Maxim Gilbert sequencing to Sanger sequencing, and then the rest was more focused more on the different generations of the NGS technology, next generation sequencing. Next generation sequencing 1, 2, and 3. So we see a good evolution of DNA sequencing starting from Maxim Gilbert sequencing to Sanger and then the NGS. Yeah. To understand it, the the application of DNA sequencing started to grow impetus due to the genomic research, diagnostics. Yeah, beyond cancer diagnostics, nowadays it is coming for infectious diseases also, and also for several of other diagnostics like several genomic diagnostics also. Yeah, so genetic diseases too. Yeah, then microbial analysis. Yeah, so to identify different microbes, we have this DNA sequencing have been extensively used. For example, during the COVID pandemic, yeah. So to understand different strains, DNA sequencing was one of the important things that came out, and as a result, people could develop a lot of diagnostics and also the therapeutics associated to it. So a lot of this depends on the DNA sequencing, and the developments of it, and also forensics. You might have seen in the several crime thrillers or crime uh, movies or such. Yeah, that how for this DNA, how it can be crucial for this forensics department. Though it is too dramatic, it shows quite dramatic, but yeah, it does have a lot of application in forensics too. So the evolution timeline starts from 1977, where Maxim Gilbert and Sanger sequencing were introduced. Next, in 2000s, NGS came in, Illumina Ion Torrent, which could give $1,000 human genome. Yeah, and then in 2010s, what we got is long read technologies. Uh, Examples of what were nanopore and PacBio. So that's how the sequencing as a technology developed, and it has been a growing field, evolving field still. So a lot of developments are still coming in, and we'll talk about the challenges too. But yeah, that's what it is. So coming to Maxim Gilbert, it's usually uses radio labeled DNA, and it uses the principle of chemical cleavage. Yeah, so it uses several chemicals, and you can essentially cleave the DNA. And then what you have to do is gel electrophoresis and radiate using autoradiography. So the pros on this technique is it is direct DNA analysis, no cloning. Yeah. And then the cons it has is it is toxic, and it can only be done for less than 500 base pair reads. Coming to Sanger, it works on the principle of chain termination. Yeah. So it has this DD piece that are being used, which stops elongation. So this uses this four reactants. Yeah, this is DDATP, DDTP, DTCTP, and DDTGP. And then you essentially use capillary electrophoresis and fluorescence detection. So the Sanger sequencing have been used quite a bit for the Human Genome Project too. Yeah, so that's what was one of the important tasks that were made successful with the sequencing technology. Coming to NGS, we have the Illumina. Yeah, so it uses flow cell clustering and sequencing by synthesis. So it is like 300 base pair reads and it's quite accurate. Then you have PacBio, which is a long read, and and then you have nanopore, essentially where you pass DNA through pores and electrical signal changes. So nanopore is also a long read technology. So after getting this sequence, you really have library preparation and QC is one of the important things. So steps is fragment DNA. You have fragment DNA of 300 to 600 base pair what you would take, and then you have adapter ligation and cluster amplification. How you can do the QC is you can analyze the fragments, yeah, fragment size by bioanalyzer and the concentration using instrument called as qubit. And then applications is you can essentially make out the non-coding regions versus the coding regions. Several other applications also will be there. Coming to bioinformatics and future, the current analysis pipeline is base calling, alignment, and variant calling. Yeah, but challenges still are there. Repetitive regions. So this is a issue with this short read technology, but it is quite okay to use long read technology like the PacBio or Nanopore, where you can also tackle this repetitive regions. So prospects is to lower the genome sequencing costs such that you can essentially go for personalized medicine that we study a lot. Yeah. So personalized medicine, we can essentially design medicines that are perfectly perfectly aligned or perfectly designed for you. Yeah. And then you can actually develop several molecular tools like CRISPR, integrate it with, and several genetic diseases can be treated as such using this molecular Caesar like technologies. Yeah. So we'll discuss the problems. Yeah. So I'll give you 5 minutes, around 5 minutes to solve the questions, and then you can write the answers in the chat, and then we can discuss it. Which key advantage distinguished the Maxim Gilbert method from early Sanger sequence? So the answer here is direct analysis of purified DNA without cloning. So uh, yeah, the Maxim Gilbert method allowed sequencing directly from purified DNA without requiring any cloning or amplification. In contrast, the early Sanger sequence required cloning to produce single-stranded DNA for sequencing, and it is one of the important advantage of the Maxim Gilbert method over the early Sanger sequencing techniques. Yeah, the answer is C. Yeah. In the Maxim Gilbert method, which reagent combination cleaves DNA at cytosine and thymine residues? So the answer is hydrazine plus piperidine. Yeah. So in cases of DMS dimethyl sulfate is something that is used for guanine. Yeah. So DMS is being used in guanine. So so this specific cleavage is what this basis is what helps in for their sequencing. Yeah. What critical structure features of DD-NTPs enables chain termination in Sanger sequencing? So the answer is absence of a 3 hydroxyl group, 3 prime hydroxyl group. Yeah. So in Sanger sequencing, DD-NTPs or D-deoxy nucleotides are used to terminate DNA synthesis. Unlike normal DNTPs, DD-NTPs lack a hydroxyl OH, yeah, OH group at the 3 carbon of the sugar molecule. This absence of the 3 prime hydroxyl group prevents the formation of prevents phosphodiester bond, yeah, with the next nucleotide, halting DNA chain elongation, yeah. This property is critical for generating DNA fragments of varying lengths, which are then analyzed to determine the DNA sequence. Yeah. So the answer is absence of a 3' hydroxyl group. Which innovation significantly improved the throughput of Sanger sequencing in the in the 2000s? So the answer is capillary gel electrophoresis. So capillary gel electrophoresis was a significant innovation which was introduced in the 2000s that greatly improved the throughput of Sanger sequencing. This method replaced traditional slab gel electrophoresis with a capillary based systems, offering several advantages. So first one was automation. So these capillary systems allowed for fully automated sample loading and detection, reducing manual labor and errors. Okay, automation. Then you have high throughput. So multiple capillaries could run in parallel, enabling processing of hundreds of samples simultaneously. So high throughput here. Then you have improved resolution. Yeah. So this capillaries provided better separation of DNA fragments, allowing for more accurate base calling, faster runtime. The use of thin capillaries reduced runtimes compared to the slab gels. Yeah, and then fluorescent labeling. Integration of fluorescently labeled ddNTPs with laser detection streamlined the process and eliminated the need for uh, hazardous radioactive labeling. Yeah. The answer is capillary gel electrophoresis. So these are the yeah, benefits. Which step ensures cluster formation in Illumina sequencing? So the answer is bridge amplification on flow cells. Yeah. So in Illumina sequencing, cluster formation is a critical step that ensures sufficient signal strength for sequencing. So this cluster formation is achieved through bridge amplification, which occurs on the surface of the flow cells. Yeah. So there are several steps. Yeah. I'll mention the steps, and maybe you can refer to it from the slides. Yeah. Adapter binding. So DNA fragments with ligated adapters bind to the complementary oligonucleotides fixed on the flow cell. Yeah. So there will be some complementary oligonucleotide, yeah, that will get fixed with the DNA with the adapters. Yeah, adapters will be there. And then amplification, the bridge amplification. So the name comes here because the bound DNA bends to form a bridge, and the DNA polymerase synthesizes and the complementary strand, creating a double-stranded bridge. Yeah, so that's the name how it comes from. Then denaturation. A double-stranded bridge is denatured, leaving two single-stranded DNA molecules attached to the flow cell. And then fourth is cluster formation. Yeah, the this process is repeated multiple times, resulting in dense clusters of identical DNA fragments, each originating from a single DNA molecule. Yeah. So, answer is bridge amplification. Yeah. So, a researcher wants to identify non-coding regulatory variants in a cancer genome. Which method is the most suitable? So the answer is whole genome sequencing. Yeah. So to identify non-coding regularity variants in a cancer genome, the whole genome sequencing is the most suitable method. If you ask why, so the whole genome sequences the entire genome, yeah, including both exons and introns, or basically coding and non-coding regions. The non-coding regions often harbor regulatory variants that affect gene expression and are crucial for understanding cancer mechanisms. Uh, the other options like whole exome sequencing. So whole exome sequencing only focus on the coding regions of the genome, which is essentially uh, less than like around 1 to 2% of the genome. Yeah, and excludes non-coding regions, making it unsuitable for identifying regulatory variants. Sanger sequencing is, you know, that is not, it is essentially limited to small scale sequencing projects and cannot handle the large scale analysis required for whole genome studies. And Maxim Gilbert sequencing is quite outdated and labor intensive and not practical for large scale genome sequencing. Yeah. So the whole genome sequencing is the correct. What limitation motivated the shift from Maxim Gilbert to Sanger sequencing? So the shift from Maxim Gilbert sequencing to Sanger sequencing was primarily motivated by the limitations of Maxim Gilbert method, which included toxic chemicals. Yeah, Maxim Gilbert sequencing requires hazardous chemicals like hydrazine, dimethyl sulfate, which pose significant safety risks to researchers. The use of radioactive leveling, like 32 isotope of phosphate, yeah, for DNA visualization added further health risk and required specialized facilities for handling and disposal. Apart from that, the complexity, yeah, the procedure was labor intensive and technically challenging compared to Sanger sequencing, which was simpler and involved enzymatic reactions rather than chemical cleavage. And low throughput. Maximum Gilbert sequencing was not easily scalable for large scale projects like genome sequencing. In pyrosequencing, detection of nucleotide incorporation relies on So the answer is chemiluminescence, yeah, from ATP. So in pyrosequencing, the detection of nucleotide incorporation relies on chemiluminescence. Yeah, signal generated by ATP. This signal is generated by ATP. So first process is nucleotide incorporation. Yeah. So a nucleotide is incorporated in the growing DNA strand by DNA polymerase, and what is released is a pyrophosphate (PPI). Yeah, PPI is released in this process. Second is ATP generation. So the released PPI, yeah, ATP generation. The released PPI is converted into ATP by enzyme called as sulfurylase. Sorry. So this PPI is changed to ATP using this uh, using this enzyme, and then this chemiluminescence signal is generated uh, using another enzyme called as luciferase. So this catalyzes a light emitting reaction using luciferin. Yeah. Then the intensity of this emitted light is proportional to the amount of ATP, which corresponds to the number of nucleotides incorporated. So the right signal is detected and recorded, which allows for real-time sequencing of DNA. Yeah. So the other options are kind of less electric current is involved in nanopore. Yeah. And ESJNJC involved in ion torrent. Yeah. Fluorescence and all are used in illumina. Yeah. So that's all. So the correct answer is chemiluminescence. Which quality control metric is critical for NGS library preparation? Yeah. So the answer is bioanalyzer. So in NGS, library preparation, correct fragment size distribution is critical for sequencing success. So the bioanalyzer is a widely used tool that provides precise information about the size and quality of DNA fragments. So this fragment size is very important, yeah. So proper fragment size ensures compatibility with the sequencing platform. Uh, and overly large or small fragments can lead to sequencing errors or reduced efficiencies, yeah. So the answer is fragment size distribution via bioanalyzer. Which technique uses zero mode waveguides for real-time sequencing? So the answer is specific biosciences. So zero mode guide, zero mode waveguides are a core technology used in Pacific Biosciences single molecule real time sequencing technology. Yeah. So this DRM-W are nanophotonic structures that confine light to extremely small volumes, enabling real-time observation of DNA synthesis at the single molecule level. So this ZMWs have very tiny aperture, yeah. So in a metallic film that limit the volume of a light excitation to zeptoliter scale. This confinement ensures that only activity of a single DNA polymerase molecule is observed without interference from the background fluorescence. So a single DNA polymerase is immobilized at the bottom of each ZMW and fluorescently labeled nucleotides are incorporated into the growing DNA strand by the polymerase. As each nucleotide is added, its fluorescence tag emits light which is detected in real time. So the answer is Pacific Biosciences. What critical role does bioinformatics play in NGS workflows? So the answer is variant calling and genome assembly. So the bioinformatics as a subject plays a critical role in NGS workflows by processing and analyzing the massive amounts of data generated during sequencing. Specifically, bioinformatics is essential for variant calling, identifying genetic variants such as single nucleotide polymorphism, insertions, deletions, and structural variants by comparing sequencing reads to reference genome. Yeah, that is calling. And genome assembly, aligning short sequencing reads to reconstruct the entire genome or mapping them to a reference genome for analysis. Yeah, so that is gene assembly. So variant calling and gene assembly, genome assembly. After an automated dye primer sequencing run, the electropherogram displays consecutive peaks of the following colors. The computer software displays the floors from DDATP as green, DDCTP as blue, DDGTP as black, and DDTP as red. What is the sequence of the region given? So red is DDTTP, which means D. Yeah. Black is DDGTP, G. Green is DDATP, which is A, and blue, which is DDCTP, which is C. So, yeah, red, black, green, A, C, G, D, A, G, C, C, C. Yeah. So that is the region given. Yeah. A DNA library shows a molarity of 12 nanomolar and average fragment size of 450 base pair on a bioanalyzer. What is its concentration in nanogram per microliter? Uh, Molecular weight per base pair. And take it as okay. Okay. So we know molarity is mass by molecular weight into volume, yeah, in liters. So here we know that it is 450 base pair, yeah. So okay, So what we can do is we can write it as 12 into 10 to the power minus 9, because it is 12 nanomolar, is equal to mass. Yeah. So we need this factor, mass by volume. Yeah, mass. Yeah. So what we can do is yeah, this will be in gram. Okay. Into 450 into 660 into we can convert this into, let it be V. Okay. That done. So basically we'll get gram per liter. Okay. So if we multiply this mass by V will come as 12 into 10 to the power minus 9 into 450 into 660. So this comes at 3.564 gram per liter. Yeah. So for converting into, nanogram per microliter, so what we have to do is 3.564 into 10 to the power minus 9 nanogram, and liter, what we can write is 10 to the power micro. Yeah, so just 10 to the power minus 6. So it will be into 10 to the power minus 6 microliter. So when we solve it, 3.564, so 10 to the power 6 here, 10 to the power 9 here, nanogram per microliter. Okay. Okay, this one maybe in a pyrosequencing readout, the graph shows peaks of luminescence corresponding to the addition of the following nucleotides: DT peak, DC peak, double height, and DT peak and DA peak. What is the sequence? So the answer is T C C T A. Yeah, that's all for today.